Okay, so I'm going to jump ship a little bit today. And even though we didn't talk about the biochem unit, pretty big part of the biochem unit, actually kind of a big part of the whole class itself is going to be enzymes. The one macromolecule we didn't talk about yesterday, which I'll bring up in a later date, is going to be the nucleic acids, which obviously is going to be DNA and RNA. Um, so again, as we go through enzymes, enzymes virtually, I mean, even then we date this back, we kind of relate this back to proteins. They have a very, very huge, major role in what we do. And probably the biggest thing that we start looking at with enzymes is just their ability to decrease the amount of activation energy or energy that is needed for chemical reactions to take place in our body. So the examples I would give you again, they're kind of like a matchmaker, and they're going to bring two substrates together that either we need to build up, or they're simply going to separate them, just depending on again what is needed. So they're going to be used in a case where you're going to break down certain proteins into amino acids, but they can also be used to actually make amino or take amino acids and build them up again. But the whole point is that they are helping to reduce the amount of activation energy, which again, if we're talking efficiency, if we're talking the overall kind of process of our body is that we're reducing the amount of energy that is needed for these reactions to occur, which we wouldn't be able to if we didn't have these enzymes taking place. So if we kind of go back to the talk with the biomolecules yesterday, remember that enzymes are proteins. Proteins are made up of amino acids, which are generally built by the ribosomes and usually dictated, not usually, they are dictated by the DNA that is found. And we can tie that into gene regulation and genes being turned on and off and everything else that we'll talk about at a later date. So again, kind of wrapping this around as far as a big picture of, the things that we talked about in class. So as we kind of look at the enzyme itself, again, remember that the shape it's taking is due to the amino acids and the R groups that you're going to find within the amino acids. And so based on the four stages of folding, you're going to end up with a molecule that can be shaped like this or any other shape as far as just based on what the R groups are. And this little active site right here is where, again, those substrates are going to bind to and either be broken down or they're going to be built back up again. Remember, enzymes end in ASE. Right, so something like DNA polymerase, all right, it's going to make DNA, RNA polymerase, reverse transcriptase, separase. All those are enzymes that are going to aid in a job or aid in a function of our body, and it's going to help to reduce the amount of energy that is needed for that to take place. Right, so again, just big picture kind of items. The big thing that we kind of start looking at, and I think we go back to are some of the graphs that we discussed with the enzymes and the lab that you guys did with the yeast and ways that you could possibly manipulate the reaction time that's occurring within the enzymes and with the substrates itself and just kind of the overall shape of that graph. Just make sure you guys are going back and kind of reviewing that a little bit as far as, when would there be a very sharp increase in the rate of reaction, eventually plateau off? Is there one that's going to be more of a steady increase and so on down the line? It's just a lot of variables come into play when we start looking at the overall rate of reaction. A lot of it has to do with just simply how many enzymes are positive, how many enzymes are there. And so if you've got a lot of a substrate and you've got very few enzymes, that rate of reaction is going to be relatively a slow, steady growth as we go up. If you've got a lot of enzymes and you've got very few substrates, it's going to be a very steep increase in the rate of reaction. And eventually, in a very short period of time, it's just going to plateau off because now there's no substrates to break down or build up and so on down the line. And so, again, a lot of variables kind of come into play with that rate of reaction. Kind of the other thing we start looking at with the actual enzymes themselves is ways that we can manipulate, we can change. And so there's actually what we call activators that you're going to find inside our body. There's inhibitors. If something is an activator, that means it's going to increase the rate of activity, basically turning it on. So we talked a little bit about chemical cellular signaling within the cells, and we'll address that again a little bit more in one of the other videos. But certain molecules, kinases, that can basically make their way through or send signals through the cell membrane can eventually work with these enzymes that can activate them by simply attaching themselves to the enzyme itself and then changing the active shape into a site that will allow it to do its function. So we've got those activators. The inhibitors work a little bit differently in that we've got two different types. You've got what they call a competitive and a non-competitive. So I'm going to scroll back up to this guy right here. So if we're going to talk about a competitive inhibitor, an inhibitor is going to slow it down. Like a toxin would be an example of an inhibitor, in that basically it's going to come in and it's going to block these enzymes and it's going to stop them from doing what they need to do. Well, a lot of that's going to be coming back to, either one, altering the shape of the enzyme, right? And the way that it could alter the shape of the enzyme is no different than an activator does, is that you've got a certain molecule that's going to come in, it's going to bind to a certain area on this enzyme, not the actual active site. And so what happens is, because it binds to it, it's going to create a different interaction within those R groups of those amino acids, and it's going to cause that active site to change. Well, in this case, if it's an inhibitor, it's going to cause it to change for the worse, and it's not going to be able to do its job. So that is what we call a non-competitive, because it doesn't compete for that active site. Right? The competitive, right, type of inhibitor is actually going to bind to that active site, and so it doesn't really change the overall shape of the enzyme. What it does is it's going to simply block that active site, so the substrates can't come in. And again, that's kind of where we start talking about some of the toxins that we find through it out of our body, or basically that we can take in, that could ultimately alter these enzymes. So a couple different ways in which they can either be activated, inhibited. Again, just make sure you guys are keeping that kind of in the back of your mind as you go on. Probably the other biggest part that we start looking at with these guys is going to be the rate at which, or the points at which they can be denatured. And so we're talking denatured, we're talking changing the shape of those active sites and the overall enzyme itself. And there's two major ones that come up. We're looking at temperature, and we're going to look at pH, acidity, salt levels. There's other things that can basically tie into this a little bit. And even though we talk about high temperature and how they can denature a protein, and high acidic environments can denature a protein, you also got to remember that we are not the only organisms that contain the enzymes. You've got bacteria that contain enzymes. You guys have learned about archaea bacteria that can live in a very, very, very harsh environment. It's high acidic, high salt, high temperature. Well, in order for them to live in those environments, their enzymes, right, have to be able to withstand those environments. Or I should say, those enzymes withstand that environment, so therefore they're able to survive in that environment. So you can see that it's not every single organism out there that is that reacts the same way to heat and to salt and or the acidity of a certain situation. And so again, when we kind of go through for human, again, right around that 98 to 99 degrees and so on is kind of where we're finding that optimal range of the temperature, or of those enzymes living inside of our residing in our body itself. And this, again, we kind of look at the pH. We have certain enzymes that will be in our pH that can alter, that can obviously live in a pH of 2, but for the most part, those that live in our body kind of need to be right around, that 7 to that 8 range in order for them to function the way that they are and not become denatured. Okay. So enzymes obviously have a huge, huge role inside of our body. Again, just kind of going back and obviously knowing what their function is, what they're doing. Again, if you see a word with ASE at the end of it, you got to be thinking enzymes. You've got to be looking at the overall function. How can they become denatured? What are ways that we can activate and inhibit them? And then also just the rate of reaction and how that can change by the amount of active sites or enzymes that are available versus the amount of substrates that are available as well. All right. Again, you guys have any questions on this video, make sure you guys are asking. Right. Otherwise, I will probably touch base with you next week. I think we'll start kind of getting into more of the cell structures, right, and go on from there. See you.